Bene, allora colleghi, come vi dicevo, abbiamo qui il nostro ospite Robert Gallo, che invito a essere qui con noi. Robert Gallo, vi faccio una presentazione, è nato nel Connecticut da una famiglia di emigranti italiani. Domenico Gallo, il nonno paterno proprietario terriero piemontese, emigrò negli Stati Uniti, come pure il nonno materno pugliese, io sono pugliese, emigrò in America per sfuggire alla povertà. Francis Gallo, suo padre, si occupava di metallurgia ed era un grande lavoratore, molto attento all'istruzione dei figli. L'ambiente in cui vive Robert Gallo è assai stimolante, infatti nel 1970, dopo l'annuncio della scoperta di nuovi virus capaci di procurare tumori e leucemie, Gallo partecipa ad un congresso tenuto a Houston da Howard Temin, premio Nobel per la medicina, sulla presenza di alcuni virus in grado di trasformare l'RNA in DNA, rimanendone particolarmente colpito. Nello stesso periodo anche David Baltimore intraprende queste stesse ricerche, così Robert Gallo decide con grande determinazione e coraggio di dedicarsi alla ricerca sui virus per i successivi 10 anni. Nel 1976 Doris Morgan, una ricercatrice del laboratorio di Gallo, riesce a far crescere in provetta i linfociti T. Ruscetti e Morgan individuano un nuovo fattore di crescita dei linfociti T, poi isolato e identificato con il nome di IL-2 interleucoina 2. Questa scoperta gli permette di isolare i linfociti T e di studiare i virus che li riguardano, come la leucemia o HTLV, primo retrovirus individuato nell'uomo. Nel 1982 Robert Gallo riceve il celebre premio Lasker per il suo studio pionieristico che ha portato alla scoperta del primo virus a RNA causa di tumori di alcune leucemie e linfomi. Il 4 maggio 1984 Gallo e i suoi collaboratori pubblicano 4 articoli sulla rivista Science cercando di dimostrare il collegamento dell'AIDS con il virus dell'HTLV scoperto dallo stesso Gallo. Così Gallo riceve il suo secondo Lasker Award nel 1986 per la determinazione che i retrovirus, oggi conosciuti come HIV-1, sono la causa della sindrome di immunodeficienza acquisita AIDS. Egli è l'unico destinatario di 2 premi Lasker. Nel 1995 Gallo pubblica la scoperta che le chemochine, una classe di composti presenti naturalmente, sono in grado di bloccare l'HIV e arrestare la progressione dell'AIDS. Questa scoperta viene proclamata dalla rivista Science come una delle più importanti innovazioni scientifiche. Le chemochine giocano un ruolo determinante nel controllare la progressione dell'infezione di HIV. Fanno comprendere meglio come agisce il sistema immunitario umano contro l'AIDS, orientando la ricerca clinica sul possibile utilizzo di una classe di farmaci per trattare l'HIV. Scusate se la presentazione è stata lunga, però credo che il dottor Gallo meriti questa presentazione e adesso lascio a lui la parola perché ci farà la sua Lectio Magistralis per quanto riguarda questa attività.
Thank you. Thanks. Well, thank you to the President. I hope you said some nice things. I'm not sure. My colleagues in biology, I will describe for you some of the work of my colleagues and myself, as I think you heard already in the introduction, that is focused throughout my career on a particular kind of cell, the T cell, and particular kinds of viruses. They have governed my entire career. So what is a T cell? As you all know, it's a lymphocyte, and a lymphocyte is a cell that we find in great abundance in the lymphatic systems, in the lymph fluid, and less so, but present in the blood. They differ from B cells. B lymphocytes make our antibodies. T cells are responsible for our cellular immune response. They produce a great variety of cytokines or interleukins which move from T cell to nearby cells that control growth, differentiation, and function of many of our blood cells, particularly in our immune responses. But if we turn the clock back to the 1960s, we only knew about lymphocytes. We couldn't discriminate between T cells and B cells. They looked the same. With the advances in monoclonal antibodies, it was possible to distinguish not only T cell from B cell, but different kinds of T cells. And then with cell sorting technology, we could begin to study their function. This was further made possible by the growth factor you already heard, interleukin-2, or T cell growth factor, that we discovered in 1976. So now we have a means to identify T cells, to isolate them, to see the subtypes, to isolate the subtypes and study their function, and to grow them for long periods of time in culture with interleukin-2, IL-2. On my first real slide, I'll tell you how this all began. Actually, before you look at this slide, even when I was a young student at 19 years old, I found myself playing with T cells. I wanted to understand the function of the thymus gland. And I made many mistakes that you could, if we had time, you would laugh. And I thought I should never go into science. But there it was, the thymus gland, T cells. I would never know that this would become later the source of everything we were doing almost in my laboratory then at the National Cancer Institute, part of the National Institutes of Health in Bethesda, Maryland, where I worked for 30 years until forming an Institute of Virology in Baltimore 17 years ago. But in the mid-1960s through the middle of the 1970s, just when I came to the National Cancer Institute, I decided to study the biology of lymphocytes, mainly not immunology. I am not really an immunologist, but the growth of lymphocytes. I wanted to compare leukemic blood cells, lymphocytic leukemia, with normal lymphocytes. Actually, how did this happen? Maybe there was a little bit of an emotional reason, because I had a young sister who died with lymphocytic leukemia at age 6. When we were studying them, we knew we had to grow the cells, but no one thought you could grow T cells from the blood. The only lymphocytes you could grow were those that were infected by a virus called Epstein-Barr virus, a herpes virus that causes mononucleosis and in combination with other factors can cause Burkitt lymphoma. So B cells could be grown if they were infected with Epstein-Barr virus. Well, otherwise, B cells could not be grown, T cells could not be grown. Many scientists thought it was impossible, in fact. It is in this period that we became aware of the work of people who made great discoveries of growth factors. For example, the work on epidermal growth factor of Stanley Cohn in the United States and of nerve growth factor with Rita Levi-Montalcini here in Italy. These factors were proteins, small proteins usually, that came out of cells and could have an effect on other cells. We exploited that observation to wonder if T cells produce such things. Well, they produce an enormous number of biologically active molecules that today we call interleukins. Indeed, when we discovered interleukin-2, it was really the first interleukin. This means a white blood cell, leukocyte, is producing a molecule that regulates another nearby cell. In other words, it's a cytokine. Not the first cytokine, maybe after interferon and one or two others, one of the earliest cytokines, but the first interleukin, even though it's called 2, that's because of the process of biology impact on T cells with antigen stimulation. Interleukin-1 has an effect before interleukin-2. Anyway, for me, It allowed us to study leukemias of these cells. My colleagues and I got into the study of viruses because of colleagues that were telling me that if you're interested in leukemia, please know that in the veterinary world of animals, leukemia is usually caused by specific kinds of viruses known as retroviruses. This was true in chickens, mice, cats, and later, after we found human retroviruses, they were also shown to cause leukemia in cows and in primates. Well, actually, cows came earlier. Primates came about the same time. In this period, we decided to develop very sensitive test systems for finding retroviruses. You may say, why? You can use electron microscopy. No one uses electron microscopy anymore for discovering viruses. Electron microscopy is not sensitive. It's very insensitive, very expensive, and requires particular technology, and very few people have access to this equipment, which is very expensive. Worse, if you do electron microscopy, you can't follow the cells every 10 minutes. But with reverse transcriptase assays, a biochemical assay, you can take a sample and every 15 minutes and measure. So what is reverse transcriptase? It's a DNA polymerase, an enzyme which catalyzes the making of DNA. But it does it from RNA to DNA, which was a unique discovery by the late Howard Temin and David Baltimore. An enzyme carried by all retroviruses, which has some very specific features. My colleagues and I spent years working on such enzymes to distinguish them from the polymerases that we have in our human cells, so we could use this test as an extremely sensitive assay, 1,000 to 10,000 times more sensitive than electron microscopy, very cheap and very rapid. Is when we culture cells, take a fraction, measure. But in this period, as we were doing this kind of work, there was a feeling strongly in the scientific community that significant infectious diseases were over. That means epidemics, pandemics. This was a thing of the past. All funding, all research should be focused on chronic degenerative diseases like arthritis and cancer and neurologic disease. The irony is that many of those diseases are caused by viruses. Cancer viruses were thought to be silly, not worth anybody spending time with. But yet, we continued this work. But I'd like to spend a moment more about this kind of attitude. After reading a little more of history, I grow to the impression that we humans have a memory of only 25 to 30 years. If you think of the great influenza epidemic of 1918-1919, which killed a big proportion of the world, if you read the history of that, the public health and scientific community was thinking that was a problem for the previous century. It had been some 30 or 40 years before there was another serious epidemic. It was another influenza of 1889. But in that century, there was also the great problem of smallpox and measles. Then came polio in the 1950s. And if you take textbooks, or not textbooks, but books of the history of polio, you will be amazed to read exactly the same thing again. At the time of the polio epidemic in the United States or Europe, scientists were saying, forget about epidemic diseases. They forgot the great flu of 1918-19, about 30 years before. So it was, again, argued, put all funding into chronic degenerative diseases. I can testify this was the case for AIDS. AIDS was first recognized in 1981 by American clinicians on the west and east coast of the United States. During the 1970s, the same sentiment was strong in America, so strong that departments of microbiology were closed in some of the most famous American medical schools. At a Cold Spring Harbor meeting organized by the great James Watson in 1974, which I attended, it was said that no human cancers were caused by viruses. Cancers were all due to chemicals or genetics, etc. In the HIV epidemic, people forgot the lessons of polio. It had been about 30 years. The Center for Disease Control, or CDC, in Atlanta, Georgia, was threatened to be closed, or at least to be reduced, because we don't need this anymore. So these are the three great epidemics or pandemics of the 20th century: the Great Flu, polio, HIV. They are all caused by RNA viruses, and in each case, it was thought we didn't have to have such worries. So the remarkable biases of the recent past is infectious diseases are over in the industrial world. Therefore, forget about them. This is a problem only for the biologists who are in the tropical disease institutes. Retroviruses do not infect human beings, and there are many reasons for this. I don't have time to detail those reasons. No viruses cause cancer in man. These biases were shattered. By the early 1980s, viruses were shown to be the cause or play a role in at least one step in cancer of 15 to 20% of all human cancer on a global scale. Now the statement is at least 20%. We found retroviruses in humans, showed that they cause some leukemias and neurological diseases, and one of the great pandemics of history, AIDS, appeared, shown to be caused by another retrovirus. I will spend a little time to tell you about these stories. Why were the prejudices so strong against even the possibility of the existence of a human retrovirus during the 1970s? One, there was decades of repeated failed attempts to find them with significant funding to do so. Second, when a retrovirus was present in an animal and causing disease, they were reproducing at very high level, making it easy to find them. However, as we noted to the critics at that time, those are the models you select to study in the laboratory. No biologist, virologist is going to study a model in which the virus is hard to find. We would soon learn that in cows, leukemia viruses exist, cause leukemia, but the virus is very hard to find. At this time, there was very little evidence in primates as disease-causing agents. That would soon change also. Number 4 is a surprise. At the time I can recall, I think it's fair to say that those people working on human disease, it was thought to be a lower form of research. Today it's very popular, it's where you get funding, it's more intellectual, it's more challenging, it's a big deal. But in the 1970s, I can tell you, it was thought to be kind of inferior research. These factors encouraged my colleagues and myself to keep on with this search. I already discussed with you interleukin-2 discovery, sensitive specific assay for retroviruses using reverse transcriptase as a surrogate marker. And conceptually, my friend William Jarrett, the late William Jarrett in Glasgow, showed that cat leukemia was spread like horizontally, infectious. Yes, it doesn't always cause leukemia. Yes, it takes a long period of time. Yes, it requires intimate contact of one cat from another. But nonetheless, an infectious cause of cancer in nature, in the wild. I mentioned already that the cow leukemia virus was discovered and replicates very little. And then we found a reverse transcriptase in a patient with lymphocytic leukemia, probably of T cells, not known at the time, and that had all the characteristics of a retrovirus enzyme. Later, the first primate leukemia virus, gibbon ape leukemia virus, was discovered. My colleagues and I discovered another variant that caused T-cell leukemia, and we witnessed an interspecies transfer, a virus moving from a gibbon ape, which is an old world Asian primate, into a new world monkey known as a woolly monkey as pets in a house of a lady. Without quite realizing it, if you look at the red on the slide, the interleukin-2 and the gibbon ape leukemia virus stories pushed me toward a study of focus on T-cell malignancies. Now, if I go back to that slide, look just above the red, the interspecies transfer. I want to spend a moment on this because it was an amazing thing, and frankly, we never really published it, and I'm not sure why. We just let it go. We were too busy at the time. That's a neck fibrosarcoma of the New World monkey, a woolly monkey, a cancer, sarcoma. And looking back, we found the woman who had this pet gibbon ape with that woolly monkey. And there's documented in the California newspaper, Sacramento Union, where she dressed up her animals on Sundays. I mean, only in California. And here's the story. He found that gibbon ape leukemia virus was present in the gibbon ape. By fighting and biting and scratching, he transmitted the virus to the woolly monkey, who developed a fibrosarcoma composed of two components, one defective virus, which has an oncogene obtained from the cellular DNA of the woolly monkey, and the other, the helper leukemia virus, gibbon ape leukemia virus. The key thing, interspecies transfer. Why should humans be an exception? In the middle years that I want to tell you about in my time at NIH was the discovery of the first human retrovirus, HTLV-1. The period is 1979 to 1980. It was first isolated from cultured growing CD4 positive T cells initiated with interleukin-2. It is the cause of adult T cell leukemia. It also causes other diseases I'll mention in a moment, especially a fatal neurologic disease. This is the first documented human cancer virus shown to directly cause the cancer with no other risk factors needed, no other cofactors needed. It's the first human retrovirus, and it remains the only real human leukemia virus. HTLV-1 is endemic in southern Japan, Australian Aboriginals, parts of Southeast Asia, South America, Iran, parts of Africa. There are descendants in the Caribbean islands in America, and it's sporadic and elsewhere, such as Europe and the United States. It is transmitted by blood, by sex, and by mother to infant, especially by breastfeeding. About somewhere 15 to 20 million people globally are infected, and it gave the idea that AIDS might be caused by a retrovirus. It also, worked with it, provided some of the key technology to find the HIV. This is the picture of the first human retrovirus, the first time we have a picture of a human retrovirus. You see a malignant T cell, leukemic T cell at the bottom, and you see the virus budding from the cell membrane as enveloped viruses are prone to do, certainly retroviruses. The dark, the electron-dense proportion is what's called the nucleus region or core region of the virus, where the genome is present, two single-stranded RNA molecules. And that's where reverse transcriptase is also located. That's the first human being from which a retrovirus was isolated. Not the first person to be infected, but this is the first time we were able to grow and isolate a retrovirus, HTLV-1. He's a young man from Alabama with no interesting history, sexual, social, personal, occupational, etc. We found the virus repeatedly in him and in several family members. Undoubtedly, he was infected from his mother by breastfeeding. The lesions you see in his skin are infiltrates with leukemic cells. This is a very aggressive disease of young adults, usually around age 25 to 45. Death usually in 1 year or less in the aggressive form of the disease, which is most cases. This is the first white person, Caucasian, And today, you don't show pictures like that anymore, but this goes back when you were allowed to do this. He had another T-cell leukemia, known as Sézary T-cell leukemia. It's the same disease with a bit different manifestations. He also had the neurologic disease, which he died of. Adult T-cell leukemia was described before the virus in 1977 by Professor Takatsuki and his co-workers in Japan, before the virus was known. I already told you, you often have skin lesions, you often have high blood calcium, and lytic lesions of the bone because one of the virus gene products induces the expression of parathyroid hormone, which causes high blood calcium and makes the lytic lesions of the bones. Lymph nodes are enlarged, and the leukemic cells, their nuclei are like this, lobulated, very diagnostic, and almost always involves the mature CD4 T cell. I already discussed some of this, which the third point is very tightly fixed in a population, almost like a genetic marker. For example, you could have an epidemic of this disease on one side of the Arno and not the other. It tends to be very difficult to transmit. And when I say it's transmitted by blood, sex, and mother to infant, it's odd. It really doesn't transmit as a virus. It transmits with whole cells. That's how poorly this virus replicates. So what happens is when there is cell-cell transmission, the donor cell makes contact with the recipient T cell, which becomes activated, and the virus can get in. So almost all infections are that way, except a hemophiliac with high level of plasma transfusions. So that means sexual transmission, cells have to cross into the bloodstream. This happens apparently with breastfeeding, and certainly among drug addicts or blood transfused people. Today, there is a required blood test. This was first developed in our laboratories. It's required in the United States. I think Brazil is endemic area for this disease. If you go to Bahia, in the hospitals, you find a lot of such cases. So it's required there. It's required in Japan, the United States. I don't know about Europe if its blood test is required today. Of course, HIV is, but HTLV is restricted where it's required. This is the global prevalence. What you see as the dominant part of the world in yellow, we don't know. It's not been tested. What you see in brown or red-brown is where we know it's most prevalent. And what you see in kind of an orange color is, maybe 0.1 to 0.8% of the population. A year later, we discovered a second human retrovirus known as HTLV2. It's not important. It's only important in that we were finding another retrovirus, so when AIDS came, what about a third one? It's also maybe important because it is the same genome almost as HTLV1, but it hardly causes disease. So understanding the molecular makeup differences between the two may give us some important information. Now, this slide is more complicated if you're not used to it, but it's only to show one thing. HTLV1 is closely related to the same virus virtually in African monkeys and chimp, whereas HTLV2 is closer to Asian monkey viruses called simian T-cell leukemia viruses, STLV. Okay? This is important only for thinking of the very ancient origin, not like HIV. This didn't come into man in the last century or so. This is thousands and thousands of years ago. The evidence that HTLV1 causes leukemia is overwhelming and probably as good as we have for the cause of any human disease. First, it's the kind of virus you find in animals that causes leukemia, often of the same type. Second, by epidemiological measures, it's very tightly linked to adult T-cell leukemia. It specifically targets mature human T cells, usually CD4 positive cells, the very kind of cells you find in adult T-cell leukemia. Fourth, it can immortalize these cells. That means in the laboratory, if I infect your blood cells with this virus, your CD4 T cells will grow forever if I worked on them carefully. And the most important is the last one. When HTLV infects a cell as a retrovirus, the RNA is converted to the DNA. The DNA integrates into the chromosomal DNA of our cells, the infected cell, and it's integrated for life. So when that cell divides, the descendants of that cell will have what we call the provirus, the DNA form of the virus. And when they divide, the next generation, and so on. That's called the provirus, integration of the sequences transmitted to the daughter cells during cell division as part of the chromosomal DNA. But in HTLV1, it's clonally integrated. This is a powerful argument from molecular biologists for causation. It means that the cell was infected at the time of transformation, not later as a passenger virus. Schematically, you can look at this slide. If you look at the right side of the slide, you see a virus that integrates into different cells randomly. It can be in one chromosome, another chromosome, one place, another place. It's infecting all those cells. But with the leukemia, once it occurs and you take the population of leukemic cells, you look on the left, your left side of the slide, you find the DNA provirus integrated in the same chromosome, in the same place, in every cell of the tumor. Now, this may vary from one patient to another exactly where, but it is in any tumor in the same place. Thanks very much. I did need that. A moment, please. And I repeat, this is about as strong an argument you could get for causation in a disease, in a cancer certainly. I'm not going to go into the molecular details. There will not be time, nor will you be very interested. But I'll give a summary of the strategy of HTLV1. The survival of the disease by viruses in general is determined by what cell they infect and their strategy to survive. So HTLV1 infects T cells, particularly CD4 T cells. To survive, when it's transmitted, it's the cell. So its survival depends on increasing, if it can, the population of infected cells. In my judgment, therefore, survival depends on inducing T-cell proliferation, and adult T-cell leukemia is the occasional accidental consequence. How does this occur? I'm not going to tell you, because there won't be time. But if this was a talk in a leukemia audience, the next 10 slides would be about that subject. Rather, and this is also, I'm not going to talk about it, and the slide should not be here. Let me go on. HTLV1 causes other diseases too. Tropical spastic paraparesis, a disease that resembles multiple sclerosis, may be a good model for multiple sclerosis. Uveitis, iritis, peripheral neuropathy, a rheumatoid arthritis-like syndrome, and modest immune deficiency. Probably these diseases are induced by autoimmunity, where the immune system is attacking its own tissues. I will move to HIV now, but I end this discussion of HTLV1 by telling you its legacy. It's the first human retrovirus, the only known leukemia virus, the only human tumor virus not known to need one or more cofactor. It can be useful in the laboratory for the immortalization of T cells. It gives us insight into the mechanisms of leukemia and demyelinating diseases, whatever their cause. And along with studies of feline leukemia virus, it provided the concept and the technology for the discovery of HIV. Those features that gave the idea are the following: the targeting of CD4 T cells, which clinicians who discovered AIDS as a disease, this showed the disease AIDS pointed out that those cells are disappearing. I've been talking to you about a leukemia where the cells are growing. Now we're seeing a disease where the CD4 T cells are vanishing. We know the HTLVs can produce mild to moderate immune deficiency. If there's a variant among them, could it cause severe immune deficiency? They originated long ago in Africa. The epidemiologists were suggesting that AIDS may have come out of Africa. They're transmitted by blood, by sex, and by mother to infant. The epidemiologists were telling us that risk factors were being transfused with blood, being a hemophiliac, etc., or by being a drug addict, or by promiscuous sex, or any sex, if you're with the wrong person, or from mother to child, mother to infant. So the hypothesis was developed between Max Essex at Harvard School of Public Health and myself that AIDS was caused by a new retrovirus, probably in the HTLV family. We predicted it would be an envelope variant. We were right, and we were wrong. This is the only theory that was productive. But believe it or not, despite all the similarities, HIV is not a brother of the HTLVs, it's a cousin, off. I end the discussion with HTLVs with a letter I received from the editor of the Journal of Virology, who was a friend, Bob Wagner. Those of you who know American stories, you'll know the stories of Dear John letters. Maybe you have them in Italy too. A soldier gets a Dear John letter from his girlfriend. It means, goodbye, I found another boy. Well, a letter of this was very kind, but it more or less said, go away. We don't want to hear about human retroviruses. They don't exist. So this is the part of that letter. This is the middle of 1980. But fortunately, it went into the proceedings of the U.S. National Academy of Sciences, and life became good for a while. So now I come to the last part of this talk. These are the middle to later years that I was at NIH, about HIV. There were wonderful years of 81 and 82, early 83. I call it the calm before the storm. AIDS is recognized in 81. CDC suggests the risk factors. Ideas of the cause are generated. These ideas included the following non-infectious causes, such as sexual stimulants like amyl nitrate, which were referred to as poppers, came out of FDA scientists and lasted until long after the evidence was conclusive, in my mind, that HIV caused AIDS, up to 88. An autoimmune reaction. This was suggested by some of my NIH colleagues. They thought this came from rough sex. White blood cells would go from the donor into the recipient. The recipient would make an immune attack, and that attack would also be on their own cells. My objection to that idea is that women have been exposed to sex, and oftentimes rough, for about 3 million years. Thus, I thought the idea was not a good idea. I hope you'll agree. Anyway, that's not the cause. So the cause also were infectious causes, many different theories. The last one on the top, the new HTLV-related retrovirus, the one I've been discussing with you, between Essex at Harvard and myself. And then I have at the bottom, ridiculous. This is particularly for the general and the lieutenant. I need help sometimes with troops. Some people say there's no AIDS. Some people convinced Mbeki in South Africa for 10 years not to give treat his people because AIDS didn't really exist, because HIV really didn't exist, or if it existed, it didn't do anything. And then there's that it's deliberately created. Why not? In the United States, of course. At NIH, where I work, why not? So, what motive? Oh, you know, whatever. So, these things are mainly gone today. But let me tell you, that it was not easy being a biologist, virologist, studying HIV in the early, mid, late 1980s. When we have a new viral infection, how does it begin? Almost always, it's a new enzootic infection. That means coming from an animal to a person, usually, almost always, by a change in society, or a recent mutation in an old known virus. Maybe that's the case with polio, for example, coming from a Coxsackie virus that became neurotropic with a mutation. influenza every season. But the society changes are critical for the thing to spread. Think back to this country. When Romans found a new port in the Middle East, new epidemic, one way or the other. When Columbus came to the New World, new epidemics, mostly for the American Indian. When air conditioning is invented, Legionnaire's disease. In my backyard, Lyme disease, we have deer. Why do we have deer? Because the society changed, the forest is smaller, etc., etc. HIV, colonialism in Africa from European powers, changing some of the dynamics, causing more people to move to the cities, famine, war, increased prostitution. You kindled the fire, which then spreads globally through sexual promiscuity and international travel greatly increasing. What are the major changes post-World War II, socially? In my mind, the increased travel, globalization, blood going from one nation to another for medical purposes. That's a big thing for the Japanese, for instance. They don't use their own blood. They took it from Europe and America. That's how they got HIV. Increased sexual promiscuity and other sexually transmitted diseases that gave rise to ulcers and other openings in the genital area that allow HIV to penetrate better. That may be one of the reasons why circumcised males are not as susceptible to HIV and are certainly less usually efficient at are more transmitting of HIV to women. So HIV came from Central Africa, it came from a chimp into man. It entered man by probably bites, scratches, cuts when a wife may be cleaning an animal for food. They're used for food everywhere, Asia, South America, Africa. I already discussed how it became epidemic. The epidemic was already spreading by the 1960s, 1970s. The transmission occurred at the beginning of the 20th century, first time. The recent static of the epidemic, 40 million people are dead, 35 million people are infected. We have a lot more to do in Africa. That's enough. What now, the very last part is now and in the future. What's the science of finding HIV and linking it to AIDS as the cause? First came the clinical description, the epidemiological information. The idea then came of a retrovirus, the technology to grow the T cells, the sensitive assays, reverse transcriptase, the first detection of a new retrovirus, then the linkage of HIV to AIDS by finding many virus isolates and making the blood test that allows screening worldwide. That's the summary. Paper of the French group was important in that it's the first report of a new virus, different from HTLV1 and 2, though a lot of people didn't think it showed enough differences. I was the reviewer. And I accepted it. It's subsequently shown in '84 to be the cause of AIDS. Its limitations. There was one isolate. The virus could not be consistently grown. The patient didn't have AIDS at that time. No conclusion was made or could be made as to its role in AIDS. Finding a virus is one thing. Showing causation is another. AIDS presented unique challenges unlike past viral epidemics or the recent SARS outbreak. First of all is the clinical latency. If someone here tonight is infected with HIV, you won't have symptoms for at least usually 4 or 5 years, 6 years, 7 years. And if that's the case, imagine when you go to a doctor or a health official, do they ask you what you did 5 years ago? Of course not. They ask what you did yesterday, last week, last month. So that's a problem. By the time a person gets AIDS, there are multiple microbial infections. Which one is the cause? These were the real key problems. When we published, we described not one, but 48 isolates of the virus. That means 48 different patients with AIDS. And in a few months after, a total of 105 isolates. But there was also additional isolates from Paris. But we still felt this would not be enough to conclude that HIV was the cause of AIDS, because verification in science is necessary. Verification by finding the virus, culturing it, isolating it would be difficult for other labs, because tissue specimens were limited. They were not even allowed inside many institutions. The ability to grow T cells, a culture technology that was not widely available in virus laboratories at this time. Third, AIDS had to be clinically recognized. And by the time the disease presented itself, when you took blood from the patient, there were very few T cells left. So very difficult. The consequence was that there were very few groups involved. In the fall of 1983, we made a major advance that seems to be not known so well. We were able to grow HIV for the first time forever in permanently growing cell lines. That made it possible for mass production of HIV. Therefore, it made it possible for a blood test globally. We could produce endless amounts. We could give it out to the world. It made it possible also to test drugs. It made it possible to do detailed molecular analysis of the genes of HIV and its proteins. It's interesting if you look at the bottom. One of the key persons in this work was my technician, Betsy Reed-Knoll, a young woman. Amazingly, while she was doing this work, her husband received blood transfusion and died of AIDS. The blood test was able to give verification of the cause because it was safe, sensitive, accurate, inexpensive, rapid, and I should also add, very specific. So verification came almost instantly when this became available. It enabled surveys of really tens of thousands of sera samples globally right away. So the major practical advances from HIV research are the blood tests that preserved the blood supply and that gave us the cause and that allowed the epidemic to be followed for the first time. You don't have to wait for someone to get AIDS. It also allowed the first screening of anti-HIV drugs, beginning with AZT in my laboratory by Dr. Sam Broder, who became the next director of the National Cancer Institute at that time. He used this very system I have just described to you. This was in collaboration with Burroughs Wellcome. This was the first time, also not so appreciated, in medical history that you have a systemic antiviral drug with proven objective decline in virus, with decline of objective findings of virus in the blood. That is, the CD4 cells go up, the virus in the blood goes down, the patient's symptoms were diminished. But it was only the beginning of therapy. It had side effects, and it wasn't that powerful. By 1995, however, with the entry of many pharmaceutical companies, we have many drug possibilities, tremendous number of drug possibilities, targeting HIV at many steps in its replication cycle. The lesson is, whereas it was thought to be impossible to have antiviral therapy because they are not like bacteria or fungi or parasites, they have no metabolism. They are just genes covered by proteins. They are intracellular parasites, obligatory. They use us to replicate themselves. How can you target them? The answer is you can. Once you know details of their molecular replication cycle, you can have more laser-type therapy, and you can have close to cures for viral diseases if you can reach it in time. Like you can with HIV, you find somebody infected early as possible, you start therapy right away now, if you can afford it. Not everybody can afford it. In my mind, the blood test is the key advance in the history of the field because it saved the blood supply and prevented further spread of the virus. It allowed the epidemic to be followed. It allowed the screening of anti-HIV drugs. we could determine who to treat, as well as block mother-to-child transmission. And lastly, it verified HIV as the cause of AIDS. This is a sidestep, only because my friend Luigi Tagliaferro told me, you should focus on key events in your laboratory. I didn't mention things related to chemokines and each thing, but I thought another virus I should mention. When we were involved in this work, I thought I was predicting things so well that I started to predict that the lymphomas and HIV infected people, which are common, they can be involved Epstein-Barr virus, but only 1/3 of the time we will discover a new herpes virus. We did. It's called herpes 6, but it has nothing to do with the lymphomas. In fact, some investigators in Japan showed it caused the infant disease known as roseola, also called xanthum sabitum, but that's the end of that story. I don't do anything with this virus. So let's talk about the needs for the future as we wind down. We need continued research on new approaches to therapy, including attempts to cure, because though people properly treated have a reasonably normal lifespan now, they still have an increased incidence of cancer and an increased incidence of cardiovascular disease. They still have to take the drug every day, but it's much, 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 much better. But we'd like to have a cure, and so there's continued research on these new therapeutic approaches. It's also true that you can develop drug resistance, so it's always good to have new approaches. The second is more important, bringing therapy to those in need, and the last and the ultimate, preventing new infections, the elimination of HIV. I turn to number 2, bringing therapy to those in need, with one slide. In the last president, George Bush, developed this program called PEPFAR, the President's Emergency Program for AIDS Relief. This is continued and expanded by President Obama. And I can tell you that it's $58 billion over 5 years. And I can tell you our institute is heavily involved in 7 African countries and in 2 Caribbean countries in the education, therapy, overall care of now about 800,000 patients. How is that possible? We do it by constantly training people from those countries. So our institute, on any day, if you would visit, you would see large numbers of people from countries, mainly the what's called the Anglophonic countries, the countries that speak English, because it's the only way we can make progress. This program needs to be expanded. It needs to survive, and I have no idea if either will occur. I don't know. It's financially difficult times. One is always worried that next year this program ends, but so far, we knock on wood, it continues. The ultimate is eliminating HIV. Education is necessary, doable, but doesn't work well. It's far from sufficient. Microbicides, there's a recent positive report from South Africa, but it requires paramedical help and doctors and nurses being available to remind the people. Microbicides means that the woman generally is inserting a protective device that includes drugs that block HIV infection. Chemoprevention means treat early and treat hard and do much more testing. Wonderful. If we do that, we'll lower the virus, the epidemic will be over. Really. It's doable. It's doable at great costs of money, of time, of effort, of willpower. I don't know if the world is able, but it is doable. I mean, that you should mark. I say it again. We have the capacity to stop the epidemic now. If we test enough, pay for it, and follow everybody and make sure they're treated right at the beginning, you lower the amount of virus. These people won't infect other people. But you have to keep following. You have to keep them on therapy. So the ultimate is the bottom. The ultimate solution is a preventive vaccine. We can all breathe free, and we don't have the financial worries. What about a preventive vaccine? The last point of this talk. There are great difficulties. One is the variation. I believe that's solvable. I believe it is being solved. The other is more tricky. Integration. We talked about it before. Let me give you the contrast. We are all vaccinated against polio, I hope. Yes? Italy, right? Let's say we take polio in a vial, a little tube, and we drink it. What happens? We will get infected. But in a few weeks, our immune system is called. We remember, we've seen something like this before because we're vaccinated. We remove it. This virus is in our gastrointestinal tract. It's an enterovirus, gastrointestinal. But polio is neurotropic. In time, it goes to the brain, but if you're vaccinated, it won't have the time to go to the brain. The virus is cleared, you don't get polio. What about HIV? Within 24 to 48 hours, it's too late. It's integrated, right? So you're vaccinated, you're going to say, oh, I just got infected with HIV, but I'm vaccinated. Yeah, sure. You better have the immune response stay high, be specific and ready, because you can't recall. You don't have the luxury to wait 3 weeks because it's over. You're infected, the virus is spreading. I hope this was clear, because this is terribly important, the point about the vaccine. That's unique. We never faced a retrovirus infection before. So one key point then already is cover the variation, make sure the immune response lasts. That's critical. Cover the variation, be powerful as your vaccine, your immunogen, and be sure it lasts. There were no, there's a fourth one, sorry. I'm out of date already. There have been 4 major trials from the United States in collaboration internationally. The first one was using the envelope of HIV. This is a good idea and a bad idea. If my hand is the virus, my fingers are the envelope. The envelope protein is then purified, my fingers. Use it as a vaccine because you'll block the ability of the virus to infect the cell. You'll make antibodies that target this envelope protein. However, the envelope is hyper-variable, greatly variable. So it only works against very limited strains of the virus. Problem. So then people turn to vaccines based on cell immunity. We will kill virus-infected cells. Too late. Too hard to cover all these things. We do need antibodies. So yeah, there is a 4th trial at the bottom. That's another failed trial. So this is our approach, and this is the end, honestly, of the talk. So our thinking at the beginning of the field, back to 1983-84, is that we need sterilizing immunity. Never achieved to our knowledge in the history of vaccines. Sterilizing immunity means you're not going to allow one virus to get through. Complete block. Okay? It's not going to be like polio. So we want to achieve or come very close to sterilizing immunity, which will require blocking HIV infection at the onset. Antibodies are likely to be required, and they should target this envelope protein. But because the envelope is so variable, standard envelope is not going to be useful. So you need to select a region of the envelope which is conserved, not variable, which is absolutely needed by HIV to infect cells and which will produce a potent antibody. The slide shows one step, and the only thing I'm going to show you, of the HIV infectious process. Note on the left of the slide that the envelope proteins, the lollipop, is interacting with a molecule on the cell surface. That molecule is CD4. It's called a co-receptor. We're back to CD4 again. When that happens, the lollipop that you see, the GP120, undergoes a major configurational change. So the envelope can get to the true receptor, which is a chemokine receptor known as CCR5, which looks like a worm going through the membrane. That's critical. So, our rationale, then we already said this. Look at the second part of this. We want to avoid targeting a site of the envelope protein known as glycoprotein 120 that have flexibility, masking by protein folding so you can't get to the key site, or coverage with carbohydrate. And this happens, actually. That's all the difficulties of getting to those sites. If such sites are critical, we believe they are, determine if you can get them exposed, and we can. How do we do this? When the envelope, my fingers again, interacts with the cell, my fist, when it hits CD4, the lollipop undergoes a big change, let's say from this to this, in order to find the gateway to enter the cell. In that sudden moment, it exposes itself to critical regions that the virus needs to be able to begin the infectious process. We'd like to get antibodies to those sites when they become exposed after binding CD4. So what are we going to do? We took the fingers and we link it to the cell CD4 molecule itself, pieces of it, the outer part. We put the two together. This changes the shape from this to this. That's the vaccine. So I don't need to tell you all this stuff. It opens this side. Let me show you graphically. The green at the bottom is the cell. You look on the left, you see CD4 as now just a stick of light green. The GP120 of the AIDS virus is coming at it. When it binds to it, I show it like an aura. That's the morphological huge change that occurs in the structure of GP120 when it binds CD4. And for simplicity, I simply show the oval GP120 on the left becoming bean-shaped, the third one in the middle. That allows it to find CCR5, the true entry into the cell. So what's the vaccine? It's the bean, the changed morphological form. So I can't go through all our results. They're in monkeys. Excuse me. But this is the red is the envelope protein, GP120. There's a linker sequence, and then the binding regions of CD4, human or monkey, depending on the experiment. Human, obviously, for human trials. And these are our results, in summary, of about 7 different experiments with maybe a few hundred different primates. There are 2 types of results. On the left is when we have a high-dose challenge, unlike a human would likely see, but one high-dose challenge. We cannot protect. You see the vaccine group in red gets infected, but we control the infection. You can see the red line is lower than the blue line, but those animals are indeed infected. They have virus in the genes of the cell. So how long would that last? I don't know. But when you look on the right side, we have sterilizing protection. This means the approach was a little different. We used the approach like a human would get, low-dose challenge, multiple times. We kept challenging the animal, and the animal's protected. Oh, it's great news. This is also a very different virus challenge from the vaccine that we use, so we can protect against a much different variable virus. So we are supported by the Gates Foundation for this vaccine, which will go into clinical trials next year, into 2014 in phase 1, then phase 2, then phase 3. I'll probably be in somewhere between here and paradise, I don't know where, by the time we get these results. It takes a long time to do vaccines. I don't recommend it for anybody over the age of 40 to get involved, really. So what's wrong? Why am I not more excited? We can't get the antibodies to last, short-lived. So we have great results for 3 to 4 months. We need to find a way to get those antibodies to last. So my betting, I have to bet, is that within the next few years, we or you will discover how to make these antibodies last longer. Somebody better, because otherwise this vaccine will be this much success when we need this much success. It'll be partially successful. but it won't be really what we want. I mentioned collaborators Tony DeVico and George Lewis in our institute, and then some people who did the animal work are below that. We have a spin-off company called Profectus. Tim Fouts was a key person in it. The Gates Foundation was enormously supportive to us. And I closed by asking if we were prepared, and the answer is yes, technically, but conceptually, we were not so prepared. And can we make sure we are in the future? We're trying. We created the Global Virus Network. It exists also in Italy now. My friend Gino Chieco-Bianchi is the head of several institutions in Italy, which are the center for Italy. We have centers in most European countries, in Asia, some in Africa. Wherever we can find enough adequate virologists, it's got to be high level. Japan, China, Russia, et cetera. So we created this global center that would say we've got to have responsible virologists available. You can't subject it to whim. My group got involved, as did the French group, as did any group that made significant strides, only by chance. I heard a lecture, maybe we should get involved and do something. We shouldn't have it up to chance anymore. I close with this. I thank you all for your attention, for the time, for giving my wife and son and myself a chance to be back in this fantastic, wondrous city, to make new friends, and a special thanks to an old friend, Luigi Tagliaferra. It's part of my pleasure of being back here is to see him and Manuela again, and I hope that he can make pasta as his mother used to made for me once. Thank you very much.
Ringraziamo il dottor Gallo per la sua dotta esposizione. Come Ordine dei Biologi, visto la rarità di avere una persona di questo livello, abbiamo deciso di porgere un segno materiale della nostra ospitalità a Robert Gallo. Gli doniamo una rarissima pubblicazione illustrata dal titolo Florence Lost, con delle illustrazioni di pregio sulla Firenze scomparsa che raffigurano scorci di questa bellissima città quando ancora erano presenti le mura di cinta, abbattute nell'Ottocento. Il testo è in inglese affinché il nostro gradito ospite possa apprezzare ancora meglio i suoi contenuti.